Welcome to our biochem review. We're going to start by taking a look at organic versus inorganic. All these compounds, they're organic. All these ones are not organic. That includes oxygen, water, and carbon dioxide. So, why are these molecules organic? They all must have something in common, and if you look at them up close, they all do contain both carbon and hydrogen. Therefore, we can make the conclusion that all organic substances contain both carbon and hydrogen together. If you're not organic, you're inorganic, meaning you don't have both carbon and hydrogen. You either have one or the other, or none at all. The organic substances, if you take fire to them, they all will burn. That includes methane, propane, butane, glucose, oil. Why are these all going to burn? The carbon-hydrogen bonds store tremendous energy. They all have carbon and hydrogen, and the bond that holds them together stores energy. Therefore, in conclusion, all organic material contain both carbon and hydrogen, and those carbon-hydrogen bonds store energy. I want to focus in quickly on water over here. It's a big misconception that water is organic. Our bodies are mostly made of water, anywhere from like 60 to 70%. Therefore, since our bodies are mostly made of water, they are mostly inorganic. Let's take a look now at 4 major organic compounds for living organisms. Our first one, carbohydrates. Probably the most delicious one, maybe in competition with fats. These carbs contain carbon, hydrogen, and oxygen, those 3 elements. And you find carbs in pasta, potatoes, rice, bread, and candy? Yup. Candy is made up of these little rings, these little geometric rings, hexagons. Those are simple sugars. And those little sugars are put together in long chains to make these big compounds that you find in pasta, that you find in bread, that you find in rice. So, if you eat pasta, bread, and rice, ultimately this pasta is going to be broken down into a big pile of sugar. So eating pasta is the same thing as eating a whole bunch of sugar. These single geometric rings or single hexagons, simple sugars, they're known as monosaccharides. The word mono refers to one, saccharide refers to sugar, because each of those rings is one sugar. Some examples of monosaccharides, the most famous, glucose, C6H12O6. Some other examples are fructose, galactose, deoxyribose, found in DNA, and ribose, which is found in RNA. If you look at the ends of all these examples, they all end in OSE. So carbs end in OSE. It's one identifier of a carbohydrate. If you look at the shapes of them, they're either a pentagon or a hexagon. So a type of geometric ring. So carbs are generally ring-shaped. A second identifier for a carbohydrate. Our third identifier is going to look at the chemical formula, C6H12O6. And if you look up close at the hydrogen and oxygen, the ratio of the hydrogen atoms to oxygen atoms will always be 2 to 1. 2 hydrogens for every 1 oxygen. And that's another identifier for a carb. So where are all these carbs or sugars made? Well, they're made by autotrophic cells, things like plants. And they're made at the chloroplasts, those little green organelles, by a process called photosynthesis. That's where the sun's energy is trapped or captured and stored in the carbon-hydrogen bonds of glucose. The purpose of making glucose or sugars is so cells could make ATP, usable energy to carry out their life functions. This occurs at the mitochondria. and it occurs in all cells, eukaryotic and prokaryotic. Eukaryotes have a mitochondria, though prokaryotic cells use their membrane. This process is cellular respiration, which again is when stored chemical energy and nutrients is transformed into usable energy for cells. How do you put these things together, these little sugars, into great big compounds? You have to remove water to do that. That's called dehydration. And if you remove water, you create bonds. You bond these glucoses together. So that's called dehydration synthesis. And the bonds that hold the sugars together are called glycosidic bonds. And they make this great big molecule of many sugars bound together, called a polysaccharide. Many sugars. There are some major examples of polysaccharides to know. The first example is how plants store extra sugar. They store it as a polysaccharide called starch, also known as amylose. How do animals store extra sugar? They make something different. They make glycogen. Glycogen is a polysaccharide by which animals store extra sugar. Plants also make the polysaccharide found in their cell walls called cellulose. Cellulose is found in plant cell walls, and plants have cell walls because they can't move. Fungi, mushrooms, and insects make a polysaccharide called chitin. Chitin is used in fungal cell walls and also found in insects' hard coats. How do you break down these great big polysaccharides? You do the opposite. You're going to add water to break down polysaccharide into monosaccharides. So hydrolysis splits. Adding water breaks the bonds and breaks down material into smaller material. Hydrolysis. Let's look at another hydrolysis reaction. We're starting off on the left with a disaccharide, two sugars together. And these are things like sucrose, table sugar is the most common one, lactose found in milk, and maltose. And these disaccharides are broken down into monosaccharides. How? What must you add to break that bond? You must add water. This process of adding water to break things down is called hydrolysis. Let's take a closer look at that dehydration synthesis and hydrolysis. This reaction shows monosaccharides coming together to make a polysaccharide. If you look in the circle, there's 2 hydrogen present and 2 oxygen. And over here, there's only 1 oxygen, no hydrogen. What must have been removed to bond these sugars together? 2 hydrogen and 1 oxygen. That's what's missing. Therefore, water was removed to synthesize this large polysaccharide. Therefore, this is known as dehydration, removing water, synthesis to build. This equation shows a polysaccharide being broken down into monosaccharides, sugars. In the circle, to start, you have no hydrogen, 1 oxygen. After it's broken down, you have 2 hydrogens and 2 oxygens. What was added? 2 hydrogens were added, and 1 oxygen was added. Water was added, and it broke the glycosidic bonds. Therefore, water will break things down. This is known as hydrolysis. Water splits. Let's take a look now at these 2 molecules. Which one has more energy? This great big one. There's more energy in this organic compound because there's a lot more CH bonds. There's a tremendous amount. This is fat, or a lipid. This little guy up here is that geometric ring. It's a sugar. Therefore, fats contain a lot more energy than carbs due to all the CH bonds. That's why you have to do so much work to get rid of fats, because you have to burn off all those CH bonds. Now we're taking a look at fats, our second major organic macromolecule. It's made of carbon, hydrogen, and oxygen, just like carbohydrates, but you find fats in nuts, cheeses, creams, and butter, different types of foods. This large molecule we identified a second ago as a fat or a lipid. There's some major examples to know. 1, phospholipids. Phospholipids make a cell membrane, all membranes in a cell. A few more examples are oil, wax, triglycerides. That word triglyceride will make sense in a second. What's the purpose of fats in the first place? Why have them? They store energy for the long term. They are also used to make membranes, as we just said before. Next question, where are fats synthesized in a cell? They're made in the smooth endoplasmic reticulum. So let's see what happens when you break down this big fat. To break it down, you have to add water. That process of breaking down a molecule into smaller molecules is hydrolysis. And fats are broken down into these building blocks, these 3 long carbon-hydrogen chains called fatty acids. Some are saturated and some fatty acid chains are unsaturated. Let's take a look. Carbon is a unique atom. It could bond to 4 things at once. When all 4 bonds are taken, it's saturated. So this shows unsaturated carbon. Still unsaturated. Now it is saturated. All 4 bonds are taken. If I remove one of these, you could still fit more hydrogen, so it's not saturated with hydrogen. It's unsaturated. So let's take a look at our unsaturated fatty acid chain. It should be missing some hydrogen. And look at that. We can put more hydrogen here. Therefore, this fatty acid chain is unsaturated because you can add more hydrogen. These fatty acid chains, nonetheless, attach to this glycerol molecule, glycerol backbone. These are the attachment points for the fatty acids. And if you want to attach those fatty acids to glycerol, you must remove water to bond them together, a process called dehydration synthesis. Now these bonds are called ester bonds. They bond the fatty acids to glycerol. These molecules, is the first one a carb or a lipid? It's a lipid, a 12-to-1 hydrogen to oxygen ratio. The second one is 2-to-1, it's carb. Hydrogen to oxygen ratio is 9-to-1 in the third, that's a fat. Number 4 is 2-to-1, that's a carb. Our third organic macromolecule, nucleic acids, made of the elements carbon, hydrogen, oxygen, phosphorus, and nitrogen. The Na nucleic acid should sound familiar. And I'm going to draw a picture to show you a nucleic acid. Now this picture shows a chromosome and the material coming out of the chromosome. The material that's coming out of the chromosome is DNA. So this material that makes a chromosome is DNA, a type of nucleic acid known as deoxyribonucleic acid. And DNA is made of a certain amount of nucleic acids, less than you think. This is 1 nucleic acid. So if that's 1 nucleic acid, how many make up DNA? 2. 2 nucleic acids in a spiraling double helix. DNA's close relative, RNA, is only made of 1 nucleic acid. RNA stands for ribonucleic acid. Let's take a look at the building blocks of the nucleic acids. Nucleic acids are made of these things, repeating units. These small pieces that make a nucleic acid up close look like this. And these are called nucleotides. There are billions of nucleotides in DNA, but only 2 nucleic acids. So, let's take a look at the parts. You notice a little circle, the geometric ring, and the rectangle. This little circle you've seen before. You've seen this in a molecule of phospholipids. This circle is the phosphate. And it's also the phosphate in phospholipids. Phosphate is just anything that contains the element phosphorus. It's a compound. The geometric ring, you know, is a simple sugar. The sugar in DNA is deoxyribose, and in RNA, the sugar is ribose. This rectangle thing is the nitrogen. It's a nitrogenous base. It's a compound that contains nitrogen. And in DNA, you have 4 ATCG, and in RNA, you have AUCG. Now let's look at our next final organic macromolecule found in meats, chicken, eggs, and fish. You find proteins here. Proteins contain the elements carbon, hydrogen, oxygen, and nitrogen. This big old thing over here is a protein. And if you want to break down proteins, of course, you have to add water. And proteins are broken down into amino acids. And there's 20 different types of amino acids. These are the building blocks of a protein. So amino acids, they're going to be bonded together to make ultimately a protein. And this happens at the ribosome. The ribosome knows how to make the protein using the DNA instructions from the nucleus. So when the ribosome puts the amino acids together by dehydration synthesis, it forms a long chain of amino acids. And those amino acids are held together by these things, these bonds, called peptide bonds. And you have to imagine there's many, many peptide bonds, hundreds and thousands of them. Therefore, proteins have an alternate name. They're known as polypeptides. Poly meaning many, peptide referring to peptide bonds. There are 4 major examples and a whole bunch more. You have tens of thousands of proteins in your body. The first one is hormones. These are chemical messages that travel in the blood. They tell cells what to do. 2 big examples are testosterone and estrogen. You also know insulin and glucagon. Next, protein enzymes, also called catalysts. The reason they're called catalysts is because they speed up chemical reactions. They speed up hydrolytic reactions, and they speed up synthesis reactions. Antibodies would be our third protein. These are made by white blood cells, and they help fight off viruses. And the hormones that we mentioned earlier, along with other messages called neurotransmitters, they're captured by receptors. These are proteins on the outside of the cell membrane that catch chemical messages. And all these proteins that we just listed, hormones, enzymes, antibodies, and receptors, if you look at the first letters of these things, HEAR, we spell out a little acronym here. Little way to help you remember them. Other examples of proteins, like the one that maybe holds the skin on your body, collagen. One that allows oxygen to travel in your blood, hemoglobin. And proteins that give color, known as pigments. The one that is most famous is chlorophyll, the one inside chloroplasts. So, what gives proteins their specific function? Meaning, like, what's the reason why proteins like testosterone and estrogen carry out different functions? Why does testosterone develop male characteristics and estrogen carry out or develop female characteristics? Two very different hormones. Let's take a look. What's the function of protein 1? It takes in molecule B. Protein 2 takes in molecule A. How did you know what the function was? The shape. The shapes match. So protein shape determines the protein function. Meaning molecule B would only go into 1 and molecule A will only go into 2. So if the shape of the protein determines its function, what determines the shape of the protein? For that, you have to look at these, the amino acids. The amino acids in 2 are the same letters or amino acids as in 1, but the order is rearranged. So the order of the amino acids is different, and a different order of amino acids will give you a different shape. So here's a little protein I'm drawing. If I take this amino acid and this one and I switch them up, that should change the shape of the protein. And it does. The shape of the protein is now changed, all because these amino acids were switched. So the order of the amino acids will determine the shape of the protein. So the order of amino acids determines the shape, and the shape determines the function. This little saying that we're writing, key importance. So that is why testosterone does different things than estrogen, because the shape, testosterone has a different shape than estrogen. Could anything influence the shape of a protein or change it? Let's take a look. Let's draw a cell receptor catching a chemical message. Right now, it works perfect. But if you apply, say, heat to this protein, something drastic will happen. The receptor does not look the same. It cannot accept the normal message that it usually did. Therefore, the protein lost its shape, and ultimately, it lost its function. So losing your shape, if you're a protein, is very bad, causes you to lose your function. When a protein loses its shape and function, it's known as a protein denaturing. This red meat, you have certain proteins reflecting red light, making the meat red, but when you cook it, those proteins lose their shape, and now they're reflecting brown because the proteins denatured. They lost their shape, and they don't reflect the same color. If you look at raw fish and you put it in citrus juices or acids, like in the dish ceviche, the fish will start to cook, meaning the proteins begin to denature. So it looks like heat and acid could cause proteins to lose their shape. So let's make a little note of this on the side. Denatures, this is when a protein loses its shape. And underneath this, we're going to write 3 factors that cause proteins to denature. So what causes a protein to denature? There are 3 things you should know. 1, too much heat, too hot. 2, too acidic. We saw that with the fish. And 3, too basic. Too much heat, too acidic, too basic. Well, if you get any of those exposed to your arm, your collagen will break down, your skin won't stay on your body. That's why burns will cause skin to come off, or acids will do the same. But when things are too cold, it just causes proteins to work slower, not to denature. That's why food lasts longer in the fridge than on the counter, because enzymes or proteins and bacteria work slower. And when proteins denature, this is permanent. You don't take a steak off the grill and put it back in the fridge and hope it turns red again, that's not going to happen. All right, so people get fevers when they're sick. Why? And why would a real high body temperature be deadly? So your body raises its own temperature ultimately to denature viral proteins or bacterial proteins. It's a defensive mechanism for your body. But sometimes your body gets carried away and it gets a little too hot. If that happens, not only will the viral proteins and bacterial proteins denature, but your own proteins, your hormones, receptors, neurotransmitters in your brain, all your proteins will denature, and that will lead to death. All right, let's look at a specific protein, enzymes. Enzymes are also called catalysts because they speed up chemical reactions. Letter A is pointing to an enzyme, and this enzyme is carrying out a reaction that starts with two things, but you end with one thing. This is a synthesis reaction because those two things were made into one thing. B and C are the reactants, the things that will go into the reaction, and D is the product, the thing that's produced. The reactants will enter the enzyme in this area. This area where the action happens is known as the active site. This is where the reactants are either broken down or put together. And the active site, you could tell, has a very specific shape, and it's made for only one very specific reaction. Therefore, each enzyme only carries out one reaction. And after that reaction, you could see the active site did not change. The shape is the same, therefore the enzyme could be used again. Enzymes are reusable. Some enzymes need some help though. This enzyme here, the shape of the active site, does not really fit the reactant at all. So, little helpers, vitamins or minerals, enter this little site on the enzyme. It's called the allosteric site. And they change the shape of the active site so the enzyme becomes functional. That's why certain vitamins and minerals are so necessary to have, because they allow certain chemical reactions to happen in your body so you can maintain homeostasis. Vitamins are organic, minerals are inorganic. Enzymes, we know they're proteins, and proteins are temperature and pH dependent. So let's take a look at this picture. What's the optimal or best pH for enzyme A? Enzyme A, the most activity happens here. Looks to be a pH of 3. The optimal or best pH is a pH of 3. Enzyme B, most action occurs here, so it looks to be about 6.3, between 6 and 7. Why does activity for enzyme D slow down here, below a pH of 11? Well, it starts to become relatively too acidic. The optimal pH is 11, and if you go away from the optimal, the protein's going to begin to denature. So a nice answer would be the relative pH becomes too acidic, causing the enzyme to denature. Enzyme B, what's the pH range? A range is from one number to another number. It starts to work at a pH of 3 and works all the way up to about a pH of 9. Therefore, it works in some capacity from a pH of 3 to 9. And the range for enzyme A works from about 0 to 5, again, in some capacity. Looking at acids, bases, and neutral solutions, this pH scale in the middle is 7, shows neutral. Anything from 0 up to 7 is acidic, and anything over 7 up to 14 is basic. Therefore, which enzyme works best in a neutral solution? Enzyme 2 has the most action at around a pH of 7. Enzyme 3, if you move it from a pH of 9 to 11, it's getting closer to the optimal pH, therefore the activity is going to increase. Always mark little dots on that graph to help you out. What happens if enzyme 1's moved from a pH of 4 to a pH of 7? Let's mark it. pH of 4 to a pH of 7. Looks like the activity decreased dramatically, so much so there is no activity because the enzyme denatured, became too basic. Let's take a look at temperature. This is a graph showing enzyme action. Letter C looks to be the optimal temperature, has the most activity. It's about a 50-degree mark. D is too hot, enzymes denaturing. A and B is too cold, but remember, proteins just work slower. Therefore, enzyme, whatever enzyme we're looking at, it denatures only at point D on this graph. Here are some examples of temperature impact. At around 70, 80 degrees, the optimal temperature for chlorophyll is fulfilled, and chlorophyll makes the green color. But when the temperature goes down, that's the optimal temperature for other proteins like chlorophyll A, which gives a yellow-green color, xanthophyll, which gives a yellow color, carotenes, which give an orange color, and anthocyanin, which gives red color. You could also see this phenomena in the summer when grass grows super fast. That's because it's the optimal temperature for the enzymes in grass to work, causing grass to grow really fast. But in the winter, grass grows slower because the temperature is colder, and proteins, all proteins, will work slower, causing grass to grow slower.